I'm excited to introduce today's speakers. Again, all three of whom are from the Environmental Molecular Science Lab, or EMSL, at the DOE's Pacific Northwest National Lab. Odeta Qafoku is the MONet campaign lead and biogeochemical transformations team lead at EMSL. She is a soil geochemist with expertise in mineral transformation at interfaces and expertise in organic mineral interactions across molecular scales using advanced microscopy techniques. Vanessa Garayburu-Caruso serves as the lead for MONet working groups and data management, and her research specializes in ultra-high-resolution mass spectrometry for environmental molecular characterization. And lastly, Arjun Chakrawal is a postdoctoral research associate at EMSL. He supports MONet through high-resolution molecular techniques such as FTICR and NMR for soil organic matter characterization, and develops data fusion tools that integrate molecular observations with biogeochemical models. This collectively advances MONet's mission to understand environmental systems at the molecular level. So I'm going to stop sharing and turn it over to the presenters. Thank you all again so much for being here. Take it away.
Oh, I know, Odeta, you are muted. We can see your slides, but unmute.
Okay, I am unmuted now. Well, I will start first by thanking Samantha and also the whole entire NEON team for this invitation to speak as part of the NEON Science Seminar Series. NEON as a program has been an inspiration because of its vision and also all the achievement that they have had. And it is an exciting moment that we will be talking here about Molecular Observation Network, or MONet, as we like to call it. Just following up on Samantha's introduction, I am Odeta Qafoku. I am one of the MONet campaign lead and also a biogeochemical transformation team lead at the EMSL at Pacific Northwest. EMSL is a DOE user facility that is supported by Biological Environmental Research Office, or BR, which as many of you know, is part of the Office of Science at the Department of Energy. What we have for you today, we have three presentations. The first presentation that I will talk about is giving a MONet overview and also touching a little bit on Soil's AI campaign. Then the second presentation by Vanessa, she will walk us through how to access MONet data and how to get involved with MONet through the working groups. And then finally, Arjun Chakrawal, he will share science highlights from MONet. So let me first start and address what is the Molecular Observation Network. MONet actually started as a user project, as a user-driven effort, with the idea of bringing the community together to build an open-source, standardized database of molecular and microscale soil data. These are data that other networks do not collect, or at least they don't collect standardized, but they are very critical if we really want to understand the processes and the mechanisms, and if we want the modelers, but also the experimentalists, to improve prediction of how soil responds to land use or energy production. MONet data is standardized, and that is what makes it powerful. And it lets us move behind single sites and start making real comparison and prediction from local to regional and even continental scales. All this effort is definitely not something that one organization can do it on its own. It only works by mobilizing the whole wider BR user community and working across sites throughout the United States. I now want to highlight how important NEON has been and the partnership with NEON has been for MONet, and to understand that we need to go through this timeline that I am showing here. MONet started in 2022. So it's a fairly new program. It's been around only for 3 years, but initially it started by launching a pilot program that was called 1,000 Soils. This was led by Emily Graham, and we, for the pilot, leaned very strongly on the partnership with NEON. And I want to mention here that NEON provided about 60% of soil samples and soil cores for the pilot project. In 2023, we launched the MONet Soil Function program as part of the user program. This was led by John Barger, and the soil function was open as a user proposal call by the MSOL website to the entire community. We saw a very strong response in 2023 by the whole BR community. And in 2024, then to keep pace with the demands from the community, we launched the quarterly soil function, where we would get the proposals every quarter that were contributing to the MONet. That's where we also continued our partnership with NEON. NEON in 2024 contributed with about 20 sites for MONet, and those sites were sites that were sensitive, such as burned, flooded, or high elevation sites that had very high value for MONet user program. In 2025, now we extended the collaboration with NEON to temporal sampling. We selected 8 different sites that, in collaboration with NEON, we selected those 8 sites that were sampled seasonally 3 times per year. I also want to highlight that in 2025, we also began shifting to community science-driven campaigns. They are focused on big challenges, and we foresee the partnership with NEON continuing also through the campaigns. Along the way, with all this progress that we did here, we also conducted core processing, soil analysis, and we also conducted 2 community science meetings in which also NEON was participating and presented their partnership with MONet. And then we also launched a searchable data portal that was called Science Central. And Vanessa will talk a little bit more about that. So how does the community participate in MONet? We made it fairly simply for community to participate. They respond to a short proposal call that is open through MSOL website, and then they do soil collection using a standardized soil kit that we send to this community. When they receive the soil kit, they take some field measurements and metadata measurements. They're pretty simple. They collect soil cores and then ship the kit to MSOL for analysis. Once the kit comes to MSOL, we do the soil processing and data analysis. And then in return, the participants receive more than 30 different data types from MONet. And I have grouped these data types in 3 major categories. The first category is biogeochemical and hydrological parameters. The second category is the high resolution organic matter characterization through FTICR measurements that are done in MSOL. And we also provide metagenome data and metagenome analysis through partnership with JGI. The third category is microscale and physical properties. Those are provided using X-ray computing tomography in MSOL, and they include parameters like pore network connectivity, soil structure, etc. Everything that you see here in green are unique data that are only part of MONet database. Now, all this that you see cannot happen without having a large team. So I want to give a shout out to some of the staff, the key staff that are generating MONet specific data. They are shown in here. There's a number of staff. And I also want to give a shout out to Emily Elofredrosh. She is at JGI and she has been a great partner over the years. So to summarize, MONet is very much about data generation. Now you've seen all the data that we are able to produce as part of MONet. It is just as important, the data accessibility. We have launched, as I mentioned earlier, Science Central with all the MONet data, and our team is working incredibly hard to make the MONet data available as soon as they are generated. It's very complex work, but we are really making great progress in this direction. Just important as the first 2 points or 2 topics, we are also focusing on community engagement and helping community use this data through training, through working groups, through support with manuscript, and help with modeling effort. Samantha has been closely involved with this engagement, and Vanessa will be talking a little bit more in detail about all the steps that have to do with accessing, and also community engagement with manuscript publication through working groups. So here is a map. Then I want to highlight that here the map is from FY22 to FY24. So we've not uploaded the newer data from 2025 yet. But you could see in this map different sites, MONet sites, and also you can see NEON sites that have been part of the MONet user program. I want to mention that through NEON partnership, we've had about 80 to 100 twin cores. And if you can calculate that, this is about 16% of our cores, of MONet cores collected up to date, have been from NEON. Here are some other statistics that are from MONet program. You could see the number of users, number of sites. Again, this is from 22 and 24, so we will update those numbers for 25. And also how many samples we have processed and analyzed, meeting participants, and also how many terabytes of data we have released. So in this slide, I would like to make one key point, and I would want to highlight here is that MONet data is not only standardized, they are structured for AI discovery. And that's what makes those data very powerful. This structure and the data standardization will let us compare data across sites, identify common patterns, and uncover mechanisms that are transferable. It also opened the door to tackling grand challenges that are bigger than one institution. And those challenges are coming to us very fast. Some of them are critical materials and minerals challenge, and also bioeconomy. And this is one of the reasons that we're moving to the community science campaign as an effort, because that is much more agile effort to address these big challenges. As we are moving into those campaigns, we are also expanding the MONet data types that we collect and will share with the community. We are adding now mineralogy, we are adding multiomics, and we're also adding high resolution spatially resolved data sets. So to describe those a little bit more in detail, they are shown here. These are all new data that we are preparing and launching for MONet. Those are the figures on the right are data that we already are producing for MONet database. In the figures on the left here, you see some new devices and new development, new capabilities that we have in EMSL, specifically terraforms. That is a way to do manipulation experiments, and also downstream analysis with these manipulation experiments. We are offering bioimaging and microscopy, and they will be all available for critical minerals campaigns that are being launched by EMSL. So this brings me to one of the campaigns where we have seen a very strong partnership with, or we have leveraged the partnership with NEON. This campaign is Soils AI campaign. And this came from analyzing MONet pilot data from 1,000 soil pilot project that I mentioned in the beginning, where as I mentioned, we had like 60% of our cores that came from NEON in that pilot project. The core hypothesis of the Soils AI campaign is that MONet data can improve prediction of carbon use efficiency and soil respiration across regions. Before this campaign was launched, when it was initially ideated, the PI of the campaign, Shakti Kumaran, he analyzed the high-resolution MONet soil organic carbon data from 1,000 Soils project and coupled this with soil properties from the open source soil survey SURGO database. And using machine learning and AI, and also the pedotransfer function, he generated a predictive map for a region of interest that is shown here in this map as a gridded map. And in each grid represent a predicted soil respiration value. While the results are promising, I would mention here that the R scale and predictability here is low. And this is because the gaps on MONet soil organic carbon data that were used for the prediction. Certain soil taxonomies were not well represented or not represented at all, and that reduced the confidence in prediction. To address this, the PI and his team, researchers from 5 universities, launched the Soils AI community campaign. The objective is to engage the broader community in collecting targeted soil data to fill the gaps in specific soil taxonomies and to improve the accuracy of the model that has been developed. So I would finalize my presentation with this slide. Again, the campaigns, the science campaigns that we are launching right now are aimed at solving big challenges. And currently there are 4 campaigns that are they are advertised in EMSL database, in EMSL website, and specific is Soils AI campaign, Soils PackQuest, Unconventional Critical campaign, and Rhizo Critical campaign. And those are the leads of each campaign. So with that, I am ready for Vanessa to start presenting her presentation.
Yes, thank you. That next slide, please. Great. So this is just a table summary of all of the data types that Odeta mentioned that MONet offers to the community. I put the link in the chat already when Odeta was mentioning, and I can put it again one more time. But this is just kind of like a quick overview of all of the different data types that are available. Next slide, please. And so as Odeta mentioned, we have launched 2 different data portals for our data types. So one is the Science Central data, which I'm going to put the link in the chat. And that's where we're storing all of the official MONet data. And then we have the Zenodo library, which is where we have the 1000 soils data. And so that is like MONet pilot. So anything that was after the pilot, so from FY23 on, it's on Science Central. Anything that belongs to the pilot, it's on the Zenodo link. And so as Odeta mentioned in her presentation, we had a very strong and great partnership with NEON during the pilot program. So a lot of the 1000 soils data is going to be co-located with NEON sites. And then with the MONet data in Science Central, we're slowly uploading some of the newer cores that Odeta showed in her timeline. So next slide, please. Here is just a quick map of all of the different MONet cores that have been collected across the different years, and then also how they pair with existing networks. So you can see that the NEON sites are the yellow and brownish circles. So we have a lot of different MONet cores collected through the years, in addition to the 1000 soils that paired with the NEON locations. So right now I'm going to give you like a quick overview of how to access this MONet data in Science Central. So next slide. The first step is to go to the Science Central website that I pasted in the chat. You're going to see there different options. You're going to click on data. Next slide. If you want to do this live while I'm talking and play around in your own computer, that could also work out. And then if you have questions or run into issues, we can answer them live during the Q&A. But basically, once you click on the data tab, you're going to see a page that looks like the screenshot, and you're going to see all data and then also a tab at the top that says MONet data. So if you click on that, then next slide, you're going to be able to see a specific search bar where you can add specific regions that you're interested for sample name. If you have a project and you know your project ID, you can add your project ID. You can put keywords from abstracts. And what this is going to do is, instead of just searching across all the different EMSL data, it's going to focus specifically to these MONet standardized data types. So for this example, I wanted us to try to just filter by NEON. So next slide. As we've mentioned, there's a lot of samples for NEON, and some of them are still coming. So if you type in that search bar NEON, you're going to see a screenshot of the map of the US, and it's going to show you kind of those density circles of where the samples are collected. And this is an interactive map. So if you're doing this live, you can actually hover over these and see how many cores have been collected, what is the core number. And so right now it shows only a few samples, but stay tuned because more it's coming. There's a big number of cores that come from neon sites. And so to get to those is the short stories, go to Science Central, MONet tab, and type for neon. Now, once you are in there, next slide, please, you can look at the right, it says your name. You should be logged in into this platform, and then you can click on the drop-down menu and hit download. And what this is going to do is that it's going to download all of the different data types that are available specifically for these neon sites that we filtered through. So if we go back to like that table that I showed at the beginning, this should include all soil biogeochemical properties, FTICR, MS data, metagenomes. If you're doing this live right now and you don't get all of these data types, it's because we're still uploading them. But the ultimate data package is going to include all of those data types that are in the table. So next slide, please. Also, the additional feature that this interactive Science Central platform gives you is that, say you have a specific research question where you're only looking at specific pH ranges in your soil type. You could go to the different drop-down menus in your interactive map and click, for example, here pH, and then play around with the range of pH observed in that red box and say, oh, I only want to download acid pHs or basic pHs. And that way the website filters that out for you when you hit download instead of downloading all of the different data types. I picked pH right here because as we're still slowly introducing more data types for these neon samples, pH was one of the ones that was available for all of them. So it was an interactive toggle that you could play with live. And so basically that summarizes the way of how to access the MONet Science Central data. For the Cenote 1000 soils data, it's just like a series of CSV files that you can download one at a time. And if you are interested in learning more, and we couldn't cover it in this presentation, we can talk more about downloading that Cenote file during the Q&A. So now, next slide, let's talk a little bit more about the MONet working groups. So like Odeta mentioned in her presentation, some of the things that we're doing within MONet is having this community engagement. We want to make sure that all of these rich data types that we are generating help you with your science. And so through this framework of working groups, our goal is to help you formulate science questions that you could answer using MONet data, help you create an action plan towards your goals, help you or your students develop data analysis skills, and then also work on manuscripts with you. And so next slide. So far, we've had two different versions of these MONet working groups. We started it first in 2024, and for these, we did a format where it was a more open format that was focused on FTIC-RMS data and biogeochemistry and XCT data. And then every month or every 2 months, we had a Zoom meeting where we invited everyone that was in the MONet listserv, either that was a participant or that was an interested person in MONet to join these lectures. And we were having kind of like a short overview about the data types and then a bit more into examples on how to use this data to answer kind of like broad and generic science questions, because we didn't want to go into details about people's specific questions and proposals, since this was just a community meeting. And so we hosted all of these online in Zoom and then recorded them and uploaded them to YouTube. Over the series of these meetings, we have over 100 MONet participants attending. And then for the practical examples where we have scripts and different data analysis tools to use these data types, we have all of these on GitHub. So I'm gonna paste the links here. The FTICR MS1 in particularly is using the 1000 Soils neon sites. And so actually Samantha was one of the attendees for this working group, and through these conversations and exploring the data types in kind of like a broad way, it inspired her to move forward with another paper that she's currently working on, that it's looking at both neon and MONet data together. And so these are a really good opportunity just to learn about how to use these data types and then connect with people and then move forward towards collaboration and papers together. So next slide. Then in 2025, we're still focusing on key MONet data types, so like FTIC-RMS, biogeochemistry, XCT, and metagenomes. And in this version, we've opened it specifically to MONet participants with active proposals or proposals that already were submitted and they have data. And our primary focus is to work on manuscripts. So we reached out to the specific MONet PIs with data and are working together with them, sometimes on a weekly, bi-weekly, or monthly basis, on how to help them achieve their goals towards developing this paper, how interacting with different MSOL and MONet experts for these data types can help them answer the science questions that inspire them in the first place to submit an MSOL proposal. And so this is a great opportunity also for grad students. If there are specific students that are looking to learn skills, we're also having working group meetings with groups of students from different labs where we teach them, for example, there is an active one, metagenomes. So that's what we're currently doing. And then next slide, please. Coming soon, we're gonna launch the 2026 MONet working group. So once again, we're going to focus on the key MONet data types, and we're going to be adding a new thing. In this case, we're going to add AI and process-based modeling. And we're going to open it to anyone, either active MONet participants with proposals and data that has been received, or any other interested researchers. So if this talk inspired you to maybe do a synthesis work between MONet data and neon sites, but you feel like you want to learn more about the FTIC-RMS or the XCT, or how to pair these data with neon data, this working group would be for you. And we're just one email away. I can paste my email in just a second, and then we can start the conversations. I'm the point person for these working groups. That doesn't necessarily mean I'm the expert at all of these data types, but I'm your point of connection for all of these. I work a lot with FTIC-RMS data, but I am not as involved in the different data types, but I will connect you with the right experts. I will coordinate the meetings, I will follow up, and we will make sure that whatever you want to get out of these working groups, you can, and that you're successful at writing your different manuscripts and producing the outcomes that you want from these MONet data, and hopefully, engage as well with the neon. And we've been working very closely with Samantha, so if you have an idea of MONet neon data, we would love to also have Samantha on board to have those conversations. So with that, I'm gonna pass it to Arjun, who's going to give you a little bit more of kind of like science highlights of how we have been using this MONet Neon partnership to publish papers and beyond, how these MONet data can be used to explore other science questions.
Thank you, Vanessa. Yeah, so thank you so much. Maybe I'll try to share my screen. Yeah, I hope you can see my screen. Okay, yeah. So thanks, Vanessa and Odeta, for providing a nice overview of MONet, its data types, and the working groups. So for me, I'm going to illustrate and present some of the science that we have done using MONet and Neon datasets. And these examples are going to be relatively brief overviews of the published or ongoing work. So if you're interested learning more about this, please look at the published papers or reach out to specific examples that hasn't been published yet. So first example is using FTICR data. So this work was led by Cheng Si and others at EMSL. So here in his work, Cheng Si hypothesized that including soil organic matter chemistry information in predictive models of soil respiration would improve the model predictive capacity. So what he did is he used FTICR data from MONet along with other biogeochemical variables collected from MONet at all of the data collected from 1,000 soils, so which also included many samples, many sites from Neon as well. So these data sets were a lot of physicochemistry variables such as total carbon, nitrogen, microbial biomass, pH, some other soil chemical variables such as manganese or magnesium ions, and FTICR data that was processed through some machine learning methods called NMF, non-negative factorization. So using these three different models, he tried to see whether including FTICR had improved the model predictions. So here we can see all of the FTICR data was processed. So if you have worked with ICR data, then you might know that FTICR has often in one soil sample, you can get more than 1,000 to 10,000 peaks representing different types of organic compounds detected. So working with such a huge complex amount of data can be somewhat challenging. So that's where this non-negative factorization comes into play. And that was the method that he used to condense that FTICR data into 7 different categories. And that's what he called it as NMF 1 to 7. So there were 3 different models that he used. One model was, so this is all, I think it was random forest or gradient boosting machine learning models. So one model where all the predictors were only soil biogeochemical variables, for example, total nitrogen, cation exchange capacity, moisture content, another model where all the features were only coming from FTICR data, and third model which combined all this information. And there was a clear increase in the predictive capacity of the machine learning model when FTIC related signatures were included in the model. So this study was like the first pass for us to understand the value of molecular scale information in predicting soil ecosystem responses. Here we used soil respiration as a proxy for that. So if you're more interested in this, there is a paper here published this year. So you can please take a look at this. And if you have more questions, you can contact me or Emily or Cheng, and we'll be happy to hear more from you. So second example is sort of following up on the work of Cheng Si, and this was led by me. So here we sort of wanted to dig deeper into the role of organic matter chemistry into trying to understand what's the mechanistic controls of this chemical diversity detected by FTICR data in uh, explaining the variations in soil respiration. So for this, I created a relatively simpler linear regression model, which included proxies for chemical diversity of organic matter and dissolved organic matter concentration that tells us about what is the easily available carbon for the microbes in soils, and some other biogeochemical variables, for example, water content, pH, temperature. So what this figure is showing you is the complex relationship that organic matter chemical diversity has with soil for predicting respiration. So let me walk you through this figure quickly. So on the y-axis, you have predicted respiration from the linear regression model. On the x-axis, you have soil respiration as a function of chemical diversity. So this chemical diversity was measured directly from FTICR data using number of peaks detected. That basically translates to number of different organic compounds which was found by FTICR measurements in one sample. So it's possible that in one sample you can have like 10,000 compounds. So that means the chemical diversity is like 10,000 for that particular sample. And here we see that for soils which had low concentrations of organic carbon. So for that, the relationship of soil respiration with organic matter chemodiversity was very different for low carbon soils versus high carbon soils. So for high carbon soils, the soil respiration decreased with increasing diversity. So soils which were chemically more diverse but had a lot of carbon, respiration tend to decrease with diversity. But for the soils which had low amount of easily available food, dissolved organic carbon, increasing chemical diversity of that organic compound, organic matter, increase the soil respiration. So with this information, there are more details about why this might happen, and I invite you to read a little bit more about this in the paper. So for now, we took this relationship and we tried to see if, so this statistical model was kind of like, okay, the chemical diversity has some predictive control over soil respiration. What if we include this information into our process-based models used to predict soil respiration as a function of dissolved organic matter concentration? So when we included this information, so that's the orange and blue lines, compared that with a model which only included dissolved organic matter concentration as a control variable to predict soil respiration. As you can see, all of these three models are pretty much, they have the same R square, so there isn't really much information gained from including dissolved organic matter chemical diversity in process-based models. So this sort of confused us in a way. So on statistical models are showing that that dissolved organic matter chemical diversity does have some predictive power over soil respiration, but process-based models said otherwise. So we kind of concluded here is that our process-based models were not capturing all the interactions that were captured by a data-driven linear regression model. So there is a room for improvement in models to incorporate FTICR data and its interactions of chemical diversity with other factors that influence soil respiration. So yeah, so the next example is on using metagenomics data that is coming from MONet. So here, this work was led is it's an ongoing work and currently under revision by Young Song. He is another postdoc at EMSL with Emily Graham. And in this work, he used the metagenomics data to understand how do soil microorganisms regulate the balance between carbon dioxide emissions and carbon sequestration. And Young processed the metagenomics data to obtain metagenome assembled genomes for all of the soil samples presented on this map here from all these sites. And here you can see a lot of these sites are co-located with neon sites. They are in fact neon sites itself. And then there are a bunch of other sites as well. And here, what he found out that you can see in this bar plot is that there is a large diversity of genome size, gene content, and other variables in the max that he processed at all these sites. And this kind of represents the widespread potential for microbial destabilization of recalcitrant carbohydrates and hydrocarbons. This working with metagenomics is not really my expertise area. So if you have any more questions, I would be happy to pass those on to Young. So please feel free to contact me or Young if you have more questions on this. And all of this data is, so if you're interested particularly working on metagenomics data that were collected from neon sites, we can help you with that as well. So moving on to one of the more focused modeling works that is being led by me and Yi Zhao. So last year we had, so PNNL has LDRD programs that was funded to build a novel model. We call this model as CRUM, chemically resolved unified microbial biogeochemistry model. So one of the main aims for developing this model was to incorporate molecular scale data from MONet and inform and some of the ecosystem scale data from NEON. So one of the key aspects of this model is that how it represents the chemical composition of organic matter. So this model is used for predicting soil ecosystem fluxes. I'm sorry about that. And here, let me quickly show you what we did. So we used FTICR data from MONet and NEON soil sensors data from NEON to inform the model for soil respiration and parameterize some of the parameters using MONet FTICR data. And here you can see three or four different lines which represents soil respiration coming out of this model comparing with other models. So let me quickly walk you through this. So on the y-axis, you have soil respiration, x-axis is the time, and in the background, it's the light blue colors, it's this soil respiration calculated using NEON soil sensor data. So for this, we used NEON soil flux R package. And this particular figure that you see is the data is coming from the Konza Prairie site in Kansas state. And what we did is we compared the soil respiration coming from our CRUM model with community land model and another soil model called is the millennial model. So the lesson learned here was that incorporating FTICR data allowed us to get the seasonal patterns in soil respiration relatively sort of similar to the matches well with the observations and also matches well with the black line which shows the simulations from community land model. But what if the differences are not that much, then what do we really gain from developing such a complex model? The gain is really this figure on the right. So all the other models, they can't really tell us about what's the source of what's the source for soil respiration. So previous models, they have dissolved organic matter represented, dissolved organic matter, or for that fact, all the pools in soil or all the pools in models representing only carbon or sometimes nitrogen. So they don't really represent what kind of organic compound that microbes are actually feeding on. And that's exactly what our models refines and the novel aspect. So here we can see five different organic compounds represented by our models that can be attributed to the total respiration. So for example, here we see that proteins and carbohydrates, the red and the blue area plots here, they contribute to the largest amount of organic to total soil respiration. So yeah, sorry. So before moving on to my summary slide, I would like to highlight that at most of these NEON sites, we also have XCT data collected. So if you're interested, please take a look at this MONet Science Center website, and you can search for these specific NEON sites. And if this data is not available, probably because they have not been uploaded yet, rest assured that we have this data. So if you want to use this in your research, we can make them available to you. So this is sort of my summary slide, which is kind of provides narrative for all the work that we have done and where does MONet and NEON partnership fits into this problem of scaling ecosystem processes. So MONet has been providing information really at molecular and core scale. And NEON, the way I say it, has information at all the scales, all the way going from sometimes processor scale to watershed and earth system scale. So I really think the real power of MONet and NEON comes from the scales represented by the two databases. MONet really gets at the molecular scale of processes, where NEON has many complementary data collected at all scales. And no one has really used this data in a combined way as before. And that's also possibly because this kind of data sets weren't really available before. So it's really up to us users of these data to ask interesting questions and make use of this wonderful resource, and also test the limits of these two databases. What kind of questions that we can ask, what kind of falsifiable testable hypothesis that we can generate using these data sets. In the few science use case examples that I presented, those are only some examples, and a lot of work has been done through MONet working groups and of partners in community using MONet data. So a lot of different work is still undergoing, and we will have more papers coming out of this using MONet data combination with Neon as well. So that's really the power that we need to investigate further. That's it. Thank you. We can take some questions now.
Wonderful. Thank you to the three of you for an excellent and rich talk. I am not seeing any written questions in the Q&A. So for everyone who's still on the webinar, if you have any questions or specific discussion topics or anything you want the speakers to elaborate on, think about putting that in the Q&A. You can also push the raise hand button, which is at the bottom of your Zoom menu. If you would like to ask the speakers a question, we can just unmute you. So I can start with one to get us going, and we do have several more minutes, so everyone think about your questions. So Arjun, following up, that was pretty interesting to think of this chemically resolved soil respiration model where it was showing the fraction, a contribution from the different compounds. I'm curious if there any way to validate that empirically? I don't know if you would have to do it with labeling studies, or do you kind of have a sense for how accurate your model might be, and how would you go about independently verifying that, or could you?
So what you said about having some sort of isotope tracer to look at what's the source of that, so that's one way to go about it. Other than that, I have not really thought about validating specifically the total respiration part of the model. Model does many things, so this is just one example. I mean, we can talk more and more. So I guess the short answer here is that I have not really thought more on what's the, other than isotope tracers, what else that we could do to get to validate the model.
No problem. Okay,
some food for thought. Something else we can think about. Okay, so we did have a question come through in the Q&A. It does say thanks for a great presentation. And then Arjun, your first science example showed how combining different data types improves prediction. The associated paper title included scaling.
Yep.
Yes. So can you elaborate what type of scaling was accomplished? What were some of the challenges of scaling these data? And maybe like what additional data could improve capacity to scale, or, think about patterns that tend to be highly variable across space and time. So
yes, excellent question, Samantha. Thank you for asking this. So by scaling here, I think what we really mean is that going from molecular, so you're trying to use molecular scale information to predict something which data at continental United States scale, for example, continental data. So soil respiration is collected at each course, but represented. So there are many samples that were representing the entire CONUS scale. So trying to come up with a way to condense this molecular scale information in order to find features or signatures that are useful to predict soil respiration. So that's one way I'm thinking of this scaling. Other is scaling of processes itself. So there might be some processes which occur at a very small process scale. So for example, interactions between microbial community and the organic matter composition. Does our data help us scale those relationships to something which might occur at a continental scale? Or so how do we, and all the data that we have here, they are all molecular scale data and some at core scale. So soil respiration, for example, was measured at core scale, but FDICI data is representative of molecular scale or organic matter chemistry. So we are already trying to relate two different scales of information. So So that's what we can think of the scaling here.
Thank you. And I kind of
had a similar question. Yeah. And Odeta, maybe you were going to speak to this, but I thought about this too in terms of the
example you illustrated where you have neon data and then you sort of try to apply it to a different ecosystem context, and
it didn't quite, I don't know if scaling in that case is right, it wasn't quite transferable. And that was sort of interesting and made me think, like, what kinds of soil diversity do we need to sample to make sure that the insights are like fully scalable to CONUS scale or fully transferable? I don't know if you wanted to address that or just say something else. Go ahead.
I'll let Odeta go first, and then I'll come back to that question, Samantha.
Yeah, I think it's a great question, actually. And I've been thinking a lot about that as well, to what can we scale. But when we look at the molecular processes, there are a lot of processes, right, that are happening in there. We're measuring all these thousands of, organic matter composition at molecular scale. But how many of those are actually really contribute to what we're measuring at the bulk scale, and even more so that the neon is measuring at field scale with the tower. So I think those are something that I don't think we have addressed yet, and will be very cool to address. And I think Arjun kind of made that connection with his last slide, where you have all these measurements at really different scale. And even in this slide, you have different scale measurements. You have molecular scale from the FTICR, but you also have bulk scale from the soil organic matter chemistry and also soil respiration. So yeah, there are scales at every level, I think. But the ultimate one will be prediction based on measurements that are and field scale with towers. I think that will be the ultimate test for everybody.
Yeah, and to continue on that, Odeta, so the scales represented by the data and the processes, linking the two has been really the challenge in soil science and ecology per se. And some of the efforts that we have been doing in MONet, in collaboration with NEON, for example, through Soils AI, some efforts in that direction. So we do need to understand whether how much of our data and our efforts can help us bridge these scaling issues.
Wonderful. All right, well, we're just about at the top of the hour, so I think we should call it there. Thank you again to our trio of speakers. This is a great talk, so interesting. We really appreciate you taking the time. The recording should be out by the end of this week if you want to go back and refer to it or share with any colleagues who couldn't make it. And lastly, we do have monthly science seminars. So we'll be back in January with a seminar on computing at the edge, also leveraging the NEON network. And so I put the link in the chat. Feel free to line up. The MONet team got so many rich links. So grab those. If those were of interest to you, reach out to the speaker team. And thanks again, everyone. Have a great rest of your day.
Thank you, Samantha. Thank you, everyone.
Thank you.
Bye. Thank you.